Bonjour tout le monde. Mon nom est Pierre Godbout et j'animerai ce webinaire. Avant de laisser la parole à la présentatrice du conférencier, voici quelques consignes d'usage. Au bas de votre écran, veuillez fermer votre caméra si ce n'est pas déjà fait. Puis, vous pouvez également activer la boîte de chat. Pour ce faire, cliquez sur l'icône bulle en bas à droite. La période de questions se fera à la fin des présentations. Et je laisse maintenant la parole à Elisabeth Gauthier.
Merci Pierre. Bonjour, je suis Elisabeth Gauthier, directrice de la division biologie forestière au Centre de foresterie des Laurentides. Bienvenue à ce 9e et avant-dernier webinaire de la 35e saison des colloques du SCF-CFL. La conférence d'aujourd'hui s'intitule « La génomique des insectes ravageurs forestiers », aussi simple que ACGT. Elle est diffusée aux intervenants du secteur forestier simultanément dans plusieurs sites à travers le Québec. Comme annoncé, le webinaire sera en anglais cette fois. C'est avec plaisir que je vous présente le conférencier d'aujourd'hui. Christopher Keeling s'est joint au Centre de foresterie des Laurentides comme chercheur scientifique en septembre 2017. Ses intérêts de recherche portent sur la structure, la fonction, l'origine et l'importance des composés naturels qui sont impliqués dans les interactions entre les plantes et les insectes ravageurs. Il est titulaire d'une maîtrise et d'un doctorat en chimie de l'Université Simon Fraser en Colombie-Britannique. Ses études postdoctorales ont porté sur la biosynthèse des défenses chimiques des conifères, la biosynthèse des phéromones chez les scolites, la physiologie des graines de conifères et le séquençage du génome du dendroctone du pin ponderosa, ainsi que de l'épidémie blanche. Il utilise une approche génomique fonctionnelle dans ses recherches, c'est-à-dire qu'il utilise les informations génomiques pour mieux comprendre la biochimie et la biologie des systèmes qu'il étudie. Je lui laisse maintenant la parole. Bon colloque. À toi, Christopher.
Merci beaucoup, Elizabeth. Merci pour votre temps aujourd'hui aussi. Donc, je vais parler en anglais maintenant. So, genomics of forest insect pests is easy as ACGT. I hope I can explain today a little bit about what are some of the things we're doing in our lab and also how we can apply genomics to forest insect pests. So what is genomics? So genomics and functional genomics and the genome are important concepts for our work. And so a genome is an organism's complete set of DNA, including all its genes. A genome does not just contain genes, but a lot of information in between. Genomics is the interdisciplinary field of biology focusing on the structure, function, evolution, mapping, and editing of genomes. And functional genomics is sort of bringing all that together so that we understand what the genome is doing in terms of the genes and the proteins and the interactions between these proteins. So at the bottom here, we have a figure which shows the three main steps of going from DNA in the nucleus of our cells to proteins such as enzymes that we use in our bodies. And so understanding this process involves understanding which genes are turned on and off in which tissues, and what do the proteins actually do that these genes are coded for. And so in the nucleus, we have our DNA, and that is transcribed to mRNA, which the M stands for messenger RNA because it carries the information of the DNA outside the nucleus so that it can be translated into protein. Pardon me. And so we've been hearing a lot about mRNA lately because of COVID. If you've been lucky enough to get a vaccine, these are mostly mRNA-based vaccines. And so what it does when we are injected by it, our body produces the antigen and we develop antibodies based on that. I'm not going to be injecting bark beetles with vaccines today and talking to you about that, but I think it's important to use the COVID story to help understand some of the concepts because compared to a mountain pine beetle or another insect, the COVID virus is relatively simple. So I'm going to discuss 3 projects today based on what we're doing in the lab. One is with bark beetles, understanding the functional genomics of bark beetle adaptations to climate and host, affecting their distribution and dynamics. And the species will be the pine beetle, or Dendroton dupin ponderosa. The second project I want to talk about is a recent project that we started on enhancing the efficacy of BTK against spruce budworm with RNA interference, and I'll explain that when I come to it. This is Tordeuse de Bourgeois d'Epanet. And also the last step is flight capacity in the Asian gypsy moth, or the spongeuse. So let's start talking about the first step, but also talking about COVID. So what are the difference between a bark beetle and the COVID virus? There's a lot of differences, but there's also a lot of similarities. So both have a host. Bark beetles attack trees, but COVID is attacking humans and maybe some other species that we don't know exactly yet. The genome size of the COVID virus is only about 3,000 nucleotides, whereas for the mountain pine beetle, it's 210 million nucleotides, or 210 megabase pairs, which I'll talk a little bit later in the talk. There's only about 15 genes in the COVID virus, but about 15,000 in the mountain pine beetle. We see variants as we're hearing the news. There's more and more variants coming out of COVID in the large population around the world. And we also see variation within the population in bark beetles. And just like we have different types of viruses that we can compare, we have different species of bark beetles which we can compare and understand, like, why are small differences causing large effects in the host that they use, or how infectious they are, and things like that. So this long line here is the, pardon me, is the genome of the SARS virus, SARS-CoV-2. It's only about 1,000 nucleotides long, and in there, these arrows represent approximately 15 different genes, and these genes code for these proteins that are involved in making up the actual virus. So the spike protein is one that is how the virus is interacting with its host. So you hear a lot about that. A lot of concern for any mutations in this spike protein may affect how well it interacts with the cell in our bodies. And so that's very similar to us and bark beetles trying to understand what changes allow the bark beetles to be more successful on their host or in a certain environment. This is a graph of the genetic variation of the COVID virus. You can see there's certain areas in the genome that have very high diversity. What that's saying is that the nucleotides and the proteins that are coding in that region have a lot of different possibilities that are being found in the populations. And so these are the concerns, the variants of concerns that we are seeing and hearing about in the news, especially in the spike protein. I think yesterday they mentioned that in India there's now a variant that has 2 of these interesting variants. And so there's a large variation in the COVID virus throughout the world. And this determines how well it interacts with its host, which in our case is us. Similarly, we can look at what's called comparative genomics. So how do these virus genomes compare with different viruses? So COVID-19 is very similar to SARS, and that's why their technical name is just COVID-1 and 2. And they have essentially exactly the same length and exactly the same number of genes. And you can see they look very, very similar. So this is what's called conserved synteny. The order of the genes are conserved across the genome. But we also have an avian virus that has very high conserved synteny as well. And so understanding and sequencing the viral genomes that infect different species and comparing them allows us to see and figure out, what is it about this that affects birds and not humans, whereas these ones can affect humans. So understanding the small differences between these similar viruses can help us understand the viruses. So from COVID, I'm going to a more complicated organism, and that's bark beetles. And so this first section is sort of an amalgamation of several different projects, but ultimately it's looking at adaptations of bark beetles to climate and their host, so affecting their distribution and dynamics. And so bark beetles typically have a 1 to 3 year life cycle, and depending on the climate, this can vary within a species. And that's important. With climate change, we may find the life cycle changes. These are very small insects in a very large forest, so they use olfaction, or their sense of smell, to find their host and find their mates. And so pheromone communication and host choice is an important aspect of their biology. They have to kill a very large tree, and so they don't do that by themselves. They use pheromones to elicit a mass attack on the tree, and they have to be able to detoxify the tree's defenses, because the tree is capable of repelling these insects with chemicals, but the beetles have adapted to these chemicals. A lot of the bark beetles also have microbial associates, and that's shown here in the blue in the galleries here. This is the blue stain fungus that our colleague Phil Tanguay is working on, among other people. And so there's also this interaction between more than just the insect and the tree, but other microbial associates. And the cold tolerance varies with the life stage. So because these insects have a life stage that goes through the winter, they must be prepared for a very cold winter. And so the insects have adapted to this process. And I'll explain that a little bit more in slides to come. So these all vary within and between species. So understanding this variation and how they differ between species allows us to predict and understand how different species may attack new hosts if their range were expanding. So this is sort of my simplified hypothesis, I guess you could say, is that a suitable climate and the availability of suitable hosts determine bark beetle dynamics and distribution. And so what determines a suitable climate or suitable host? Well, to a large part, it is the genome and the natural variations within the population that determine what climate and hosts are suitable. And the variation allows the beetle to adapt to a changing environment. I could also add that the tree, of course, plays a role too. It is an organism with a genome and natural variations, and so that has a role as well in this process. So sequencing genomes from several bark beetles will allow us to study these processes in more detail and start to get a better understanding of these processes. So I'm been working on 4 different insects for genomics. The mountain pine beetle, which I hope most of you know about. It is a beetle of Western North America, but now it's progressing eastward into the boreal forest, and there's a concern that it will move through the jack pine of the boreal forest, either coming through Alberta into Saskatchewan, or potentially through human-aided migration from the Western United States into the Midwest here. So this is an insect of great concern, and it attacks pine hosts. We're also looking at the spruce beetle that attacks spruce, and it's found across the range of spruce in Canada, but particularly at the moment in British Columbia, it's a problem. And so this is an interesting species as well. The Douglas fir beetle is found only on Douglas fir, which grows on the west coast, and it again is another dendrogenous species. And finally, the western balsam bark beetle. This is a beetle that attacks fir or abies, and it has a big role in the subalpine fir in British Columbia. And so what we have here are 4 different species. 3 of them are in the Dendrophilus genus, and western balsam bark beetle, the Dryopsites genus. They all are eating different genera of trees: pinus, spruce, Douglas fir, or fir, and they all have slightly different life cycles. Mountain pine beetle is typically 1 year cycle, spruce beetle is 2, Douglas fir is 1, and western balsam bark beetle is 2. So you could imagine possibly with climate change, these cycles may change faster or slower depending on a warmer or colder environment. And this can be important for the population dynamics of this species. So this is a very messy slide, and it's part of the problem with genome assemblies is that we incorporate a lot of different data to try and make the genome as best as we can. And so for people who do genomics, I've included this slide, but what we're showing here is that we start off with in our case, we have in mountain pine beetle a genome assembly that we did in 2013, and we're trying to improve that with various steps. And for the other 3 species, we started off with a draft assembly using a technology called 10x Genomics Chromium. And so with further steps, we improve and improve the assembly to get our assembly, final assembly. And actually, this is showing here that I think his name's Ponko. He's the mascot for the Potholes of Montreal. And this just reminds us that everything is always improving or hope to be improved. And so even the human genome is still improving as we get more and more information. So in example, we received this week some additional data that we're going to be incorporating into these genomes to make them even better. So this is the table of the statistics of the current state of the assemblies. And I just want you to look at a couple of different columns. So over here, we have what's called the karyotype. That means how many chromosomes are in the genome. So this, in the mountain pine beetle, it has 11 autosomes and the sex chromosomes. And the size of this genome is 208 megabase pairs. So that's 208 million base pairs. And so we compare that to, I want to compare that to things. Here, this number means 90% of the assembly is contained in 12 pieces. So that compares very well to our karyotype, which has 12 pieces as well, just including the Neo-X in the case of the female. And so what we're finding here is that we have essentially all the chromosomes as individual pieces, although we have a bunch of little small pieces left over that we have still to sort of find the right place for. The other thing I want you to point out to you is the sum. So this is the sum of all these pieces is 214 megabase pairs, and that's very similar to the 208 determined through another means. So this tells us that for the mountain pine beetle, we've actually got a very, very good assembly. There's still a few little straggling pieces, but generally we have a chromosome level assembly. This is not quite so good for the spruce beetle, Douglas fir beetle, and western balsam bark beetle. We see here that 90% of the assembly is contained in 10,000 pieces. So that's why we're still incorporating more data to improve on these assemblies. Okay, that's enough for that table. We now have these chromosome size scaffolds, and just like I showed you for the COVID virus, the whole genome was one piece. This is actually just one chromosome. Chromosome 10, mountain pine beetle, is over 3 million bases long compared to the 30,000 in the COVID virus. And each one of these blue marks here are different gene. We now have identified all the genes in the mountain pine beetle genome, about 13,000, and for spruce beetle about 15,000. We're still working on the other 2. So now we have this grand catalog of all the genes in the genomes. And the question is, what do we want to do with it? And it depends on who you are and what interests you. But we, of course, are interested in the processes of post-colonization and dynamics of the populations. So that's what aspects we're looking at. But it's just an encyclopedia of this insect. And so depending on one's interest, we can study other aspects of it. So with the COVID virus, I mentioned conserved synteny, and we see conserved synteny as well between the different bark beetle species. So these graphs are called Jupiter plots, and they're split down the middle, left to right. And so on the left side here, we have the scaffolds or the chromosomes of the mountain pine beetle female. And here are the smaller pieces. These are the first 12 I mentioned, and then some of the smaller pieces. And over on this side is the spruce beetle male. And you can see some of them are chromosome-sized pieces, and then we have some smaller pieces at the top. And what you can see here is that these bands that go across represent how similar those two pieces are on the two species. And you can see, for instance, let's say, look at this one, they're very similar. All the genes are in a line on one chromosome on the one species are very similar on the other species. But we also see changes here where things are crossing over. And this suggests that between these two species, the genomes have actually been scrambled a bit between the two. And that may provide an advantage to one species over the other for various reasons that we could explore. Similarly, for the Douglas fir beetle, we see quite high synteny, conserved synteny between the mountain pine beetle and the Douglas fir beetle. So this is one way we can look at the bark beetle species, compare them on a very large scale, a genome-wide scale. But we're interested in some of the specific processes, and that includes adaptation to cold. Beetles must survive Canada's winters, and they do so by a couple of processes. Lowering their freezing point with cryoprotectants. So just like the antifreeze we put in our car, insects make antifreeze so they don't freeze. And they also synchronize their life cycle with the seasons so that they try to make the most cold, hardy life stages endure the winter. And we'll talk about that in just a sec. Work is in collaboration with Kathy Bleicher and Greg Smith at Pacific Forestry Center. They are biologists and research scientists looking at cold physiology and diapause. And so I'm collaborating with them to incorporate this genomic component. They've shown that the species of bark beetle, the eggs and pupa are least resistant to cold, and late instar larvae and adults are more resistant to cold. They also have different freezing points depending on the life stage. And so the insects use a process called diapause to help synchronize their development with the environment. So this is a schematic of the life cycle of mountain pine beetle. An egg is laid by the female in the summer, and it develops through the first instar, second instar, third instar, fourth instar during the early fall. But then it decides its winter is coming, so it goes into diapause. And diapause is a programmed response to those conditions, and the suppressed development can last longer than the conditions. This is different than quiescence, which is immediate response to adverse conditions. The way I think about this is if you find a spider, let's say, in your house, you catch it and put it in the fridge, that spider is going to quickly quiet down and stay quiet in the fridge until you take it out again. But as soon as you take it out and it warms up, it will move along again. So that would be quiescence. Whereas diapause is a process that the insects may warm up, but they will still be in this quiet state. And an example that I experience here in Quebec City is the ladybugs that come into our house in the fall, and they actually diapause inside our house in the cracks in our walls. You don't see them in the middle of the winter, but come springtime, you often see them starting to come out again. That's because these ladybugs are diaposing in our house. So even though it's relatively warm in our house, they don't come out until they're ready. So mountain pine beetle is a 1-year life cycle. Once the fourth instar goes through the winter and it's now spring, it develops into a pupa, and then a tenoral adult becomes an adult, and the cycle repeats itself. Spruce beetle has generally a 2-year cycle. So it starts out similarly, but the overwinters at the fourth instar larva and diapauses. Then the second year, it sort of doesn't do a lot. It goes from fourth instar larva to pupa to tenoral adult, but then it must diapause again as this tenoral adult, and it spends a second winter as an adult before it finally emerges as a full adult. Now, with climate change, or if you are on a warmer side of the slope versus a colder side of a mountain, it can vary its life cycle. So with climate change, if it's a warmer climate, this can be shortened to 1 year. So it develops all the way up to a tenoral adult before winter, diapauses as a tenoral adult, and then comes out the next year. Or if it's colder, it doesn't get a chance to develop far enough, so it stays maybe a third instar larva over the winter, develops the second year to the fourth instar larva, and then diapauses, and the cycle continues. So you can imagine that there's sort of there's limits on what this insect can do. If it develops too far and develops into a pupa just before winter, that pupa may die over the winter. And so the insect is trying to coordinate the seasons with the life cycles that it is most apt to survive the winter. So we're looking at this through gene expression analysis. That's so looking at the levels of mRNA or messenger RNA in the insects. Right now we're looking at well, sort of 3 processes. One in mountain pine beetle for this fourth instar diapause, but also in spruce beetle at both diapauses. And so I just want to briefly talk to you about this second diapause, where the tenoral adult spends winter as the tenoral adult develops into a full adult, emerged adult in the spring. So what we're seeing, and I'm going to explain this figure in just a second, is that there's significant differences in the transcript levels of many genes in tenoral adults before and after diapause. So this figure here is called a heat map, and each column represents one individual beetle, and each row represents one gene in the genome. And what we have colored here is that these are all the samples before they went into diapause, and these are samples that went like after diapause. And you can see here, based on the coloring, that some genes went down in expression levels, so I'm going from orange to blue, but then others went from low levels to high levels, or low levels to very high levels. So we're exploring this. This only shows 50 genes, but we have approximately 15,000 genes here that we can look at, and we can explore what are these genes that are changing when the insect goes through diapause, and how does this process work. Another aspect that we're looking at is the adaptations to host. So when the beetle emerges from its natal tree and starts flying to try and find a new host and conspecifics, they recognize that the host is a host and not a non-host. They have this pheromone-mediated aggregation to overcome host defenses. And you can see that here, where each one of these little dots on the tree is where a beetle has gone in, attacked, and the tree has tried to push them out by releasing chemicals. the beetles have to tolerate these host chemicals and eventually overcome the defenses of the tree. And these processes are challenged when a beetle expands its range, maybe through climate change, and into a host that it's not used to. So this is exactly what's happening in Western Canada, where the mountain pine beetle is now entering jack pine, which both have not seen each other before. So one aspect of this is olfaction. So the insects must smell their hosts and their conspecifics. And the process of olfaction involves many different types of genes. And so we can now go into the genome and identify these genes. They're all there. We just have to explore and find them. And these differ between the different conifer feeding species. So I've been collaborating with some colleagues in Norway and also in Sweden to look at these in both mountain pine beetle as well as Ips typographus, which is the spruce beetle in Europe. So we can start to compare between the different species. The aggregation pheromones are also very important for bark beetles, and it's quite a complicated process, and there's several pheromones involved. And these are some of the mountain pine beetle pheromones. So we can start to look in the genome and identify and postulate how these pheromones are biosynthesized and try to identify the genes involved in these processes. And these papers show some of the examples where we'll find the genes involved in these processes and showing how they work. And we also are looking at how the insects tolerate host defenses. As we've shown, and others have shown in the past too, mountain pine beetle, it has a certain level of mortality when it's exposed to these compounds. And so they must respond by trying to detoxify or avoid or tolerate these compounds. And this is done through enzymes called cytochrome P450s. Those are much like the enzymes in our liver that break down medicines and things like that. We also have found something interesting happening, that the larvae are actually modifying these compounds and adding a fatty acid to them and actually carrying them along as an adult. And this is part of actually their pheromone process. And that's in our lab. Kathy Bernier is looking at an enzyme here, and you may have heard her talk in the fall at Cafe Science. So we were able to look into the genome and identify some of these enzymes that are working on these processes and functionally characterize them and show how they work. And this allows us to understand better how these insects are able to tolerate these toxic compounds. So in summary, genomes of 4 important conifer bark beetles have been sequenced. The Genomics supports research to understand the biology, biochemistry, and population genetics of bark beetles. We are currently, in addition to improving the genomes, we're also looking at those genomes comparatively and continuing our functional genomics studies on diapause and host colonization. And later, once we understand these a little bit better, we can start to look at the populations and look for variants and how those variants may affect the dynamics of the populations of these different beetles. So next, I want to talk about a different species, and that's the spruce budworm. This project has been funded by Surge International last year, and also this coming year we also have Surge funding and also a little bit of A-based funding. And this is to enhance the efficacy of BTK against spruce budworm with RNAi, or RNA interference. And this is everybody, well, not everybody, but all the involved are at the center here. So people in Quebec for sure certainly know more about spruce budworm than me. I'm from the West Coast and not had much experience with the spruce budworm, but I'm getting into that. But the eastern spruce budworm Christinura fumiferana is actually found on the East Coast, or sorry, Eastern Canada mostly, but there's different species. It's a species complex of Christinura that is found all across America. And so it's an interesting genus to study because there's different hosts, different life cycles, and different processes to explore, and they all have a certain level of pest problems. So spruce budworm, pardon me, has been controlled in the past using DDT, and this, you probably are aware of, is no longer being used because it's a chlorinated hydrocarbon with major environmental impact. It tended to affect non-target organisms, so like eagles, and it would bioaccumulate. So that was banned, I think, early 70s. Since then, BTK, or Bacillus thuringiensis, I can never say that right, thuringiensis kurtzsaki, is a bacteria that contains a toxin that is specific to Lepidopteran insects. And this is very effective. This can be sprayed as well. And one of the disadvantages of this is that it's not species specific. And so if there's, for instance, endangered or threatened Lepidoptera, which there are several in Canada, one should avoid spraying this BTK near there because that would kill those insects as well. And so that's a little bit of a problem in some areas. But there's also the potential, just like any pesticide, for resistance by the insects. And there is the potential for BTK resistance. A postdoc in Michel Cousson's lab, Sandrine Pink, has shown that the variation in the gene involved in BTK resistance in other moths is also found in this species. So we don't actually see BTK resistance yet, but the potential for the population to become more resistant is certainly not impossible. So the idea of thinking about other means of controlling these insects is certainly on my mind. So one of the techniques that is becoming popular, especially in agriculture, is what's called RNAi, or RNA interference. And I'm just going to walk you through this. I know this text is too small probably for you to read, but I don't want you to read it. In the normal cell on the left side here, the DNA, as I mentioned at the beginning, is converted into mRNA, and this mRNA is used to make the protein in the cells. And so this is the normal process that's going on in cells. Now, if we were to introduce double-stranded RNA into the cell, and so double-stranded RNA is two pieces of RNA that are bound to each other, similar to DNA, but it's actually RNA. This is also called dsRNA. When it goes into the cell, there's an enzyme in the cell that cuts it up into smaller pieces, and this binds into a protein complex, which then binds to the mRNA that is complementary to this small piece. So this nucleotide sequence that is here would be the exact same sequence here. And what this causes is that this causes this mRNA to be destroyed and chopped up. So if we design double-stranded RNA that is complementary or matches an important life-giving sequence for the insect, we're going to chop that up, and it will no longer make this protein, and potentially that insect is going to die. And so this has become a very interesting process, especially in agriculture, for controlling pests. So we can make it very specific because genomes, no two genomes are alike. Pick a region of the genome that is very unique, that is affecting a very important gene in that insect, and it could potentially be very species specific, and it will not affect other organisms that don't have that nucleotide sequence. In agriculture, they're using, well, genetically modified plants to actually produce double-stranded RNA in the plant. They can also spray the double-stranded RNA onto the plants, and there's various means of doing this. So we're looking at doing this with spruce budworm. So our objective is to develop an RNAi interference-based biocontrol agent to enhance BTK spray products for spruce budworm, and we want to target the genes important for survival and development. Perhaps RNAi will improve the efficacy of BTK at current spray concentrations, or maybe we'll be able to reduce the spray concentrations. And as we learn more about the process of RNAi and its utility in pest management, we may be able to replace BTK spray with an RNAi spray in the future. The advantage of these RNAi processes, just like you were hearing in the news with COVID vaccines, is that because it's based on nucleotide information, if that nucleotide information changes, for instance, if there's a variant in the population, it's relatively straightforward to change that piece of DNA or RNA and make a more effective target. So this allows us to improve the efficacy should there be a change in the population dynamics or other processes. And it's also good for changing to a different organism as well. As long as we have the genome, we can predict what sequence we can use. So our project involves 3 steps, of which we're only going to work on the first 2 until we've had good success. We're going to identify in the laboratory candidate genes, and test them using double-stranded RNA and sublethal doses of BTK. And then once we've found some that work really well, we're going to test them in the greenhouse with spraying. And maybe in several years' time, if this is successful, we would like to try doing experimental plots. That's a long ways away, but it's something that we hope to accomplish in the future. We can produce the RNAi in the lab. There's 2 usual steps, an in vitro step, which means we synthesize this double-stranded RNA based on the DNA sequence, or we make it in vivo. So we're using E. coli, a special strain of E. coli, and a special plasmid that will produce this double-stranded RNA, and then we can kill the bacteria containing this double-stranded RNA and feed it to the insects or even spray it. This is some of the techniques that they are using in agriculture. We are targeting important developmental genes, and we're using the precedent from agricultural pests to help us choose which genes are most potent in agricultural pests. And now, because we have the spruce budworm genome, which Michel Cousson and others are working on, we're able to go in and find the same gene in spruce budworm that has been effective in some agricultural pest. So again, we're relying on the genome here for information. We expose the insects in one of several ways. Currently, we are doing microinjection, but we could feed them a little droplet of the double-stranded RNA or put them in artificial diet. We then observe the larval development and survival. So this picture here shows what we are doing when we do an injection. We have this micro-injection device. It's just a syringe on a little motor that is very precisely controlled. And you can see here, sitting on the ice, on this blue square is a 4th instar spruce budworm larva right there. And this blue solution inside the needle is our double-stranded RNA. We color it blue so that when we inject it, we can see that the color changes in the inside of the insect so that we know we've properly injected them. Because we want to make sure that these are sequence specific and species specific, we are going to look for non-target insects and how does our double-stranded RNA affect them. And we're choosing the cabbage looper and the white-marked tussock moth. Both of these now have genomes available so that we can confirm that the sequence we choose is not in these genomes. And this is actually a project by Daniel Doucette and Amanda Rowe at Great Lakes Forestry Center. They've recently sequenced the white-marked tussock moth as well as the forest tent caterpillar. And so those genomes will soon become available for other kinds of studies as well. We've just started this RNAi project, and so I don't have any results to show you per se, but the results are to come. The last project I want to talk about is the flight capacity in the Asian gypsy moth. This is a very collaborative project, part of the Canada-funded BioSafe project. So we're looking at the ability of female Asian gypsy moths to fly. Now, for those not familiar with the gypsy moth, this has been an introduced species into North America for, I guess it's over 100 years now. There are two main strains of gypsy moth. The European strain, which the female does not fly, and then there's the Asian strain where it can fly. So in between zone or introgression between these two types. So in Eastern North America, there is the flight incapable strain, and this has been slowly percolating in the States and in Canada for many years. But there's a concern, especially with global trade, that this flight capable go across the and establish itself in Western North America. And there's been several examples of it coming there, but we've been able to wipe it out. They're attracted to the light, and you can see the, like a CFIA nightmare where all these gypsy moths are attracted to the light and they're laying masses. And so the boats have protocols to try and clean these up before they get into Canadian waters or close to port. But the CFIA would like to know, when they see a moth on a ship, is this European and therefore not flight capable, or is this a flight capable Asian moth? And so we're in the larger BioSafe project developing tools to allow the CFIA to be able to detect whether this is a flight capable or flight incapable insect. So they can have, the level of risk increases when it's a flight capable insect. So this project involved 3 analyses of gypsy moth flight genetics, a GWAS, which was analyzed by Gwilym Blackburn, who's now at the Pacific Forestry Center, inbred lines by Julian, and gene expression by me. And I'm only going to be talking about the gene expression. We were looking at gene expression in gypsy moth pupa. So our assumption is that the flight capacity of flight capacity of adult females developed during the pupal stage, and we should be able to see these differences in development through gene expression. And so what we have is the 2 strains. The orange strain came from a flight capable location, and the CT strain came from the incapable location. And we collected 5 pupa for each of them at day 1, 3, 5, 8, 11 during the pupal stage. And with a gene expression analysis using a process called RNA-seq. And here we were using the Asian gypsy moth genome as a reference, and this was a paper that came out by our collaborators at the University of Laval. So this is a local genome project as well on the Asian gypsy moth. So what we see is, as you would expect, there would be quite a few changes in the gene expression just because of different populations. So we see between the Russian population and the population from Connecticut, there's quite a few genes that are up or down between the 2 strains. We also see, because as you can imagine, the pupa is developing with time through the pupal stage, a lot of the genes are changing, going both up and down during the pupal stage. But what we were looking for is those genes that are changing differently with the development between the 2 strains. And so that's sort of the interaction between the day strain. And there's actually only 170 genes that were significantly different between these interactions. And and so here is the heat map for this interaction. This is the top 50 genes. And what you can see here is in the blue and the red are the strains at the 5 different days. And if you sit back and look, you can see some glaring differences. So for instance, here at day 8, the flight-capable strain has much higher expression for these genes than does the flight-incapable strain. And similarly down here, and this opposite pattern where the flight-incapable strain has higher expression than the flight-capable strain. So we're looking at all these different genes that are significantly different. And what we're finding is that many of them are involved in cuticle development. And you would expect that flight capacity is probably controlled by many genes. It can be a behavior, it can be muscle, it can be cuticle, but a lot of them seem to be cuticle development that we're seeing. And some of these genes are known in other insects. And for instance, in Drosophila, they show patterns when they are mutated, they show patterns of difference in their ability to fly or their patterns in their wings. And so I think we're on the right track in finding some of these genes that may be involved in flight capacity in the Asian gypsy moth. And so in order to test that, we want to functionally characterize them. And so the way we plan to do that is to, once we've got the spruce budworm working for RNAi, we want to use the spruce budworm as a proxy for the Asian gypsy moth. The Asian gypsy moth is kind of not desired to be in the lab in Canada for obvious reasons. So we want to use the spruce budworm and try and use RNAi to prevent the spruce budworm from flying by targeting these same genes in spruce budworm. And so we hope once we have got the RNAi worked out in spruce budworm, to work with Lucy Roy and her staff to study the flight capability of spruce budworm that has been affected by RNAi. So finally, in summary, I just wanted to acknowledge that the genome assembly projects provide important resource for many projects. I've shown you several projects that have built upon genome assemblies, and this is just those within sort of my sphere of collaboration, but these genomes go out and are publicly available and anyone can use them for whatever reason they're interested in. I know for instance, the mountain pine beetle, there's people in Germany that are looking at the digestive genes in the gut of mountain pine beetle to see if these can be used for commercial industrial purposes. There's a whole slew of different applications for the genomes of these insects because they have adapted to these very unique environments. And genomics is not just a means unto itself, it supports and complements the research to understand the biology, biochemistry, population genetics of insects. So, you know, what we're learning out in the field can be enhanced and sometimes help to be explained through the use of genomics. And also, genomics can improve our ability to monitor and manage pest insects. So with that, I'd like to acknowledge the many people and funding organizations that have been involved in this over a number of years that I've been working on these things. Those that are underlined above are people in the Canadian Forestry Service, but we, as you can see, it's a very collaborative, multi-institutional, multi-disciplinary project that involves genomics in forest insects. Thank you very much for your time. Merci beaucoup. Et,
Alors,
okay.
merci Christopher. Bonjour à tous. Je suis Pierre Godbout et j'animerai la période de questions. Vous pouvez maintenant activer votre vidéo si vous le désirez, en bas à droite. En bas dans le milieu, je veux dire. Comment procéder pour poser une question? Alors, vous pouvez poser votre question dans la langue officielle de votre choix, avec le micro de l'ordinateur, en cliquant à droite de votre nom dans la liste des participants et sélectionnez l'icône main levée. Lorsque je vous nommerai, vous n'aurez qu'à enlever votre mode sourdine en cliquant sur l'icône micro. Et après avoir posé votre question, s'il vous plaît, vous remettre en mode sourdine. Vous pouvez également utiliser la boîte de chat si vous préférez poser votre question par écrit. Alors, sélectionnez l'icône en forme de bulle en bas à droite pour l'activer si ce n'est pas déjà fait. On vous demande de vous identifier clairement en donnant votre nom et le nom de l'organisation que vous représentez ou pour qui vous travaillez. Alors, la période de questions est maintenant ouverte et je vais donner immédiatement la parole à Madame Nathalie Isabelle, qui a levé sa main.
Bonjour, Christopher. Merci beaucoup pour la présentation. Il y a beaucoup de projets dans ton laboratoire. C'est vraiment intéressant. What I would like to know is when you compare the different species of beetle, have you also have a look at their hosts in terms of species divergence and which traits could be also be involved in the host insect specificity, like the phenology, or what do you know about those?
Yeah, so as you know, we're just developing the genomes of conifers as well. There's white spruce and some pines and things, and so there is certainly the interaction between those. And so we're taking it sort of one step at a time. So for instance, you know, looking at the conifer defenses, the different species have a different diversity of defenses, and the different species of insects have to combat those. And so comparing the tree defenses between species and the way that the insects combat them is part of that interaction. Yeah, for sure. So as I mentioned, the cytochrome P450s, there's a group of almost 100 different P450s involved in bark beetles, and they may diverge between the different species based on the divergence of the conifers as well. So it's important to, yes, keep the conifer in mind. Certainly. So that's part of the interesting aspect and why we chose all 4 species of beetles having a different host species, genera, I should say.
Okay, thank you.
Yeah.
Alors, il n'y a pas de questions dans le chat. Je rappelle aux gens, s'ils veulent poser une question, vous n'avez qu'à lever la main en allant dans la liste des participants. Et à côté de votre nom, vous allez avoir une petite main levée qui va apparaître. Sélectionnez-la. Et on a un commentaire, une question dans la boîte de chat que je vais lire. Donc, c'est Kishan Sambaraju, qui est chercheur au Centre de foresterie des Laurentides. Hi, Chris. Great presentation. You mentioned about diapause in the mountain pine beetle. However, literature doesn't suggest obligate diapause in mountain pine beetle during the winter. Can you talk more about this?
Yeah, I would prefer to divert that to Kathy Bleicher, who's been studying that more directly, but my understanding is it's more a facultative diapause rather than an obligate diapause. So they don't have to go into diapause to complete reproductive development, for instance, but it's more facultative, which means if the conditions are right, they would do it. Does that answer your question? Certainly, depending on the species, it's either facultative or obligate diapause. And so that needs to be pulled apart and decided for each species in particular.
Okay. Donc, il te remercie. Est-ce que d'autres questions? Okay, Kishan? Yeah. Go ahead.
Yeah, right. So the literature doesn't suggest obligate diapause, and it's the larvae that have like, you know, you talked about the antifreeze compounds that are produced during the winter. So I'm not sure there is either, well, obviously there is no obligate diapause, but even facultative diapause hasn't been proven yet. It hasn't been published. You might know that paper by Benz that talks about pre-pupil diapause. So that area, I think it needs more work.
And in fact, Kathy Bleicher is actively working on that. So she has done some work to suggest that this is probably what's happening. And some of the experiments that I'm doing with the RNA-seq builds upon her work.
Okay, thanks.
But I can just mention that it is a difficult situation to pull apart because the insects are developing and also adapting to the cold. So setting up the conditions for the experiment to test these is often quite difficult. How do you differentiate between a facultative and obligate diapause and things like that is sometimes quite critical.
Yeah. So is it is it pre-programmed? Is it like by December does it go into diapause just by a little like a indication of cold? Does it go automatically into diapause, or does it?
So we're looking in that, so we're looking at, whether it's a factor of light cycle and or temperature. So if the temperature is below a certain temperature, or if the daylight hours is less than a certain amount. So both of those can have an effect on whether they enter diapause or not. So I'm still exploring that.
Okay, thanks.
Je rappelle aux gens pour une dernière fois, je crois. Alors, Nathalie, vas-y. Je vois que tu as ta main levée live.
Oui, l'autre n'a pas l'air de fonctionner. J'ai posé sur le bouton, puis ça fait une barre ou ça fait, je ne sais pas.
Pas de trouble.
Okay, Christopher, I would like to know, in terms of RNAi approach, are you aware about other systems where they use it, and like in the wild, or is it like something that is going on only in lab situation, or was it already used also in other for other type of insect than spruce budworm?
So certainly RNAi is used in a lab as sort of a research tool to understand the function of genes, and that's quite widespread. I've used it myself in bark beetles and such, but to use it as a pest management tool is becoming more and more common, but especially in agriculture. So I don't have all the examples in mind, but there's, for instance, a genetically modified potato that produces a RNAi for the Colorado potato beetle.
Okay.
And so when the insect eats the plant, it ingests these RNAi and then dies. And so I don't think genetically modified organisms is the best thing to do in a naturally managed forest. And so the idea of spraying what would be a dead double-stranded RNA, it's not live, it's even less, I would say maybe less benign than BT, or more benign than BTK, would be a better approach. And so the spraying process is also being actively studied in the agricultural setting as well. And so, how do we incorporate the double-stranded RNA into sprays for good efficacy, for long stability and things like that? And so there's certainly a lot of literature developing for this process.
Okay. And what is the social licensing when those approach? I know that for agriculture, people are more willing to test the new approach, but even if there is still some there's debate a lot, but when it's the time when you speak about natural forests, it's totally another, forests are considered as sacred, and it means that you cannot touch them. So have you already started to talk with people and to do perhaps some to co-develop project with different community, and I would like to hear you about it, because it's something that you need to come at the beginning of the process, not at the end. So,
well, we haven't yet too much, because we want to in the lab show some efficacy, because I agree that the regulatory and public perception hurdles are huge to introduce a new technology into the forest for pest management. But I think that is necessary to collaborate with people, social scientists and things, to gauge the response.
yeah.
Personally, I think in some ways, when fully understood, double-stranded RNA would be more likely to be accepted than even BTK. BTK is less species specific. It is a spore that has a certain viability, whereas double-stranded RNA is dead. And it can be made appropriately to be very species specific. So there's certainly some advantages to double-stranded RNA than to BTK, and we hope that the Canadian government regulatory system will help to appreciate some of the advantages and disadvantages as it becomes more applied in agricultural as well. But personally, I'm very cautious about trying to introduce something into a natural environment without thorough testing and regulatory oversight. But I think we want to show that it works first.
Okay, thank you.
Yeah.
Je vois pas d'autres questions dans le chat. Pas d'autres mains levées. Nathalie, une dernière chance? Bon.
No, I think that I discover actually what is good. It's when you see the presentation of your colleagues, sometimes you realize that they do different things where you can connect. And I would like to know, perhaps my last question is, how do you are you working also with the modeler to look at also at I would say more at the population effect for, because we know that for a given species that there might be like a local adaptation. I know it from the tree perspective, but I'm pretty sure that this is something also for insect, and some insect might go from one species they can move to another host. So is this something that we need to also to be aware of for the mountain pine beetle and other beetles, spruce beetle?
There's certainly a lot of genetic variation in the insects, and so how that manifests in what hosts it attacks and its success in colder environments, things like that. It's very open to further understanding. Some of my colleagues at the center, for instance, Deepa Kiraswaran and others, have studied, for instance, the pheromone composition of spruce beetle across Canada. And so there's actually quite a large difference in the components and the relative amounts across Canada. So we definitely see differences there, and that could manifest itself in how well it can mass attack trees and things like that. But in addition, these variations within the species may affect how successful they are on different hosts. And so
Okay.
We do not have a very good data set yet of genetic diversity in spruce beetle, for instance, across Canada, but that's something we want to do in the future. And then we can start looking at some of these questions where they're on different hosts and different locations.
Don't forget to collect the DNA from the host.
Yeah, it's always a complicated process, but this is what's so neat, is that it's the tree interacting with the insect and interacting with all the microorganisms under a certain environment, and these are all changing.
Yeah, thank you.
Merci.
Bon, je crois qu'on a fait le tour des questions. Je regarde dans la boîte de chat, puis les mains levées, il n'y a plus rien. Donc, merci Christopher pour ta présentation. Génomique des insectes ravageurs forestiers, aussi simple que ACGT. En terminant, je vous invite au prochain et dernier colloque de la saison 2020-21 qui aura lieu jeudi le 8 avril et qui s'intitule « Lutter contre le marché noir d'un produit vert ». Les conférenciers seront Nathalie Isabelle et Danny Rioux, chercheurs scientifiques de Ressources naturelles Canada, Service canadien des forêts, Centre de foresterie des Laurentides, et Isabelle Duchesne, chercheure scientifique de Ressources naturelles Canada, du Centre canadien sur la fibre de bois. Surveillez également notre fil Twitter et LinkedIn pour en savoir plus. Merci à tous de votre participation et bonne fin de journée. Merci Christopher.
Merci Pierre. Bonne journée à tous.